Okay, let's go ahead and get started. So I'm going to start with that chapter 12. I do have your assignment, so that will help me stay on track. Right? So I'll start here. Chapter 12. Once upon a time, we started with nervous tissue, right? Because we had 4 different types of tissue, right? Nervous, muscle, connective, and epithelial, right? 4 general types of tissue. Nervous tissue was its own kind. Nervous tissue does not regenerate like the other tissues. It's completely different. It's unique, right? So in terms of nervous tissue, we're talking about the functional unit of the nervous system, which is what? The neuron, right? The neuron, functional unit of the nervous system. Simple question, right? What is the functional unit of the nervous system? Neuron, okay? The neuron is considered to be an excitable cell because it responds to electrical changes, just like the muscles, right? The neuron itself is a cell, and it's composed of a soma, right, and an axon. The soma is usually going to be unmyelinated area that contains a nucleus and the Nissl bodies. The Nissl bodies are made of rough endoplasmic reticulum with ribosomes, and that's where you're going to synthesize your neurotransmitters and other stuff too, but so inside neurotransmitter because that's what we're talking about in the nervous system mostly, okay? The soma also receives input via extensions known as dendrites, right? Dendrites. So you have soma, and you have your dendrites, and you have your Nissl bodies. Okay? From there, it's going to communicate with the axon via the gate known as the axon hillock, right? Which is the gate, or anatomically to me, it looks like the neck of that neuron. Okay? The axon is the tail, and what it does is allows communication over long distances. Okay? So that axon can be unmyelinated sometimes, or myelinated, depending, right, on what we're talking about, or myelinated. So generally, when we're talking about gray matter, we're referring to soma, and when we're referring to white matter, we're talking about those axons because of the fat. Okay, so just to generalize those 2 terms. This axon is going to transport action potentials to a synapse and also transport neurotransmitters to the synapse. The flow of information from the soma to the axon is known as anterograde. Anterograde because it's going down the line, it's going forward, right? It's improving. Now, because the center of the axon is a cell, it has mitochondria, it has stuff in it, it produces waste, right? Because mitochondria are doing metabolism, and from metabolism, you get CO2, you get waste, right? So that waste is transported back to the soma, so it can be digested, and that would be retrograde transport, okay? So those are the main 2 parts of the neuron. Now, in terms of neurons, we talked about different kinds of neurons. I'm just going to put the ones that we talked about the most, because that is what we're going to see the most. And you're going to have here the multipolar neuron, multipolar, which is the most abundant, so multipolar for the most. And then you're going to have these bipolar neurons, and the bipolar neurons are B for brain senses, so you're going to have it in your special senses, like your smell, like your eyes, like your ears, okay? So special senses there, okay? Let's see from there. So let's move on to the support cells, right? Support cells. What were the support cells generally called? Neuroglia, right? Or glial cells. We have different support cells in the central nervous system than the peripheral nervous system, right? The central nervous system neurons, neuroglia, right? The first one, let's start with myelin production. Who produces myelin in the central nervous system? Oligodendrocytes, right? Who produces the cerebrospinal fluid? Yeah, ependymal cells. Okay? Who produces or makes up the blood-brain barrier? Astrocytes. And who does phagocytosis? Microglia, yes. Okay? What is phagocytosis? Eating, cell eating. Okay, in the peripheral nervous system, we only had two, right? And that was Schwann, right? Everybody loves the Schwann cells. The answer I saw the most on the test, on the lab test. Everyone's like, Schwann everywhere. Like all 20 spaces says Schwann cell. So Schwann cell, which is what? Myelin, right? Myelin in peripheral nervous system. And then you have the other S for satellite. And the satellite cells were support cells in the sense that they controlled different things, gas exchange, neurotransmitter production, etc. So support cells, the satellite cells are usually wrapped around the soma. Usually wrapped around soma. And the Schwann cells are wrapped around the axon. Okay? So those were the basic cells of that nervous system. All right. In terms of neurons, we also have three different types of neurons when we're talking about the circuitry. Can somebody pronounce that for me? Like when you have a circuit and then circuitry, right? All right. So you're going to have the sensory portion, sensory neurons, and those are what? Afferent, right? So this is peripheral nervous system. You're going to have the interneuron, and the interneuron is integration, and that is usually central nervous system. And then you're going to have the motor neuron, which is efferent, right? E for exit, efferent, which is again peripheral nervous system, right? So you have those three main classifications of neurons. Okay? You did that? You did that? Okay. So, any questions on that? That's just vocabulary words, right? So let's go ahead and go into the everybody's favorite topic, right? What's everybody's favorite topic? Take a guess. The action potential, right? The action potential. One day, this is like sometimes things, right? They just click one day it's just gonna slap you on the face and you're like, oh, action potential. It just happens that way. Okay, so right here, we're gonna put the charges, right? -70, -55 millivolts, and then at the top here, +30 millivolts, and down here, -90 millivolts. We're talking about the action potential, we're talking about the depolarization of the membrane, right? So we're gonna write things down. This would be depolarization, this would be repolarization, this would be hyperpolarization, and this would be the polarization, which also means resting membrane potential, right? So resting membrane potential equaled having a salty banana, right? Salty banana. have your potassium on the inside and your sodium on the outside. And the general charge is estimated to be negative 70 millivolts when that is in balance, right? So the action potential, we talked about it being all or nothing, right? All or nothing. So what we're focused on in this chapter more is this little piece right here, which is your graded potential, right? The graded potential is known as a local membrane change, right? Local membrane change. They can be, or sometimes, most of the time, they're sub-threshold. So sub-threshold stimuli. By themselves, they cannot cause an action potential. So they're considered generally to be sub-threshold. What is sub-threshold? Less than what? Less than negative 55 millivolts, okay? Meaning that a stimulation, if I'm resting at negative 70 and I get stimulated, it might stimulate it to negative 60. Not enough for an action potential. It's all or nothing. You either make it or you don't. Like your driver's test, right? You get a 69, you don't pass. You have to get a 70. But once you get a 70, it doesn't matter what grade you get, right? The person that got a 70 is driving with the person that got a 90. So defensive driving, right? All right. So graded local membrane changes, they are sub-threshold. They occur at the soma, right? At the soma of the neuron. And the soma is the one that is going to decide if that membrane potential is enough to go down the axon, right? That's why I drew it curvy like that, right? Because you have two types of graded potentials. You're going to have either depolarizing graded potentials or hyperpolarizing gradient potentials. Okay? So let's talk about the first ones here. Depolarizing. Depolarizing equals moving towards threshold. In other words, making the cell, the neuron, less negative. I was going to say more positive, but let's say less negative, okay? Less negative, okay? So it's bringing it closer to the negative 55. So moving towards threshold is depolarizing. Cell is less negative. So these are going to be your excitatory neurotransmitters, right? Which we also known as EPSPs, right? In terms of those excitatory neurotransmitters, you're going to have some that are for the central nervous system and some that are for the peripheral nervous system, and sometimes they're different, sometimes they're the same, but let's generalize right here. In the central nervous system, you have things like glutamate, and glutamate is the main excitatory neurotransmitter of the central nervous system. In the peripheral nervous system excitatory, you're going to have acetylcholine into nicotinic, or you can have norepinephrine, epinephrine, right? So you have those neurotransmitters, they're going to bring the cell closer to that threshold. You can also have the hyperpolarizing graded potentials, right? And those make the cell more negative, which makes it go further from threshold, right? So it's harder to stimulate. They're putting you further away. So further from threshold, we are going to call those IPSPs, which are inhibitory. In terms of those inhibitory neurotransmitters, for the central nervous system, we talked about GABA, and in the peripheral nervous system, we talked about acetylcholine into muscarinic receptors. Okay? So tying it all together like that, right? I was going to say one more thing. Okay. Moving towards, I want to put the number. Moving towards threshold, and I'm going to put here negative 55. And then more negative, negative 90, right? So those are how you get to that threshold. So why did I do the squiggly line? Because you can have both stimulatory and inhibitory neurotransmitters being deposited into that neuron at the same time. So we do something with graded potentials that is called summation, like in math, right? Adding. So we have summation, we're going to add them together. Okay? So summation, and we did have two types of summation. We had temporal summation, which was due to time, and we had spatial summation, which was due to space. Okay? So how did that work? In temporal summation, you're going to have, like, for example, one neuron releasing these neurotransmitters, not one at a time, but over a short period of time. So this is like somebody knocking on your door over and over again, right? Acetylcholine, 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 acetylcholine. Eventually, you're going to get up from the couch and open the door. Okay? So it's over time, just one at a time, or somebody poking you, right? First time they poke you in the back to ask you about, what's answer number 5? What's the answer number 5, right? The first time you're like, did I hear that? But then the fourth or fifth time you're like, stop. What time? Right? All right, spatial summation is multiple neurons firing at the same time, right? And in this case, you could have like a neuron secreting acetylcholine, and then this one, let's say it's GABA, right, inhibitory, and then acetylcholine again. And what we're going to do is we're going to add and subtract from them, right? So if I have more excitatory, let me just put excitatory here and inhibitory instead. EPSP, IPSP, EPSP. If the overall net result is excitatory, then maybe we get to threshold and pass that action potential. But it's multiple neurons secreting neurotransmitters into that postsynaptic neuron at the same time. So this is like multiple people knocking on your door at the same time. You hear a loud boom, you're going to get off the couch versus like the solar person. Like, you're like, somebody knock, right? Now, in temporal summation over time, you have to do it in quick succession. It has to happen rather quickly, one right after the other. Because imagine if you're at home and somebody comes in at 6 a.m. and knocks on your door one time and then leaves. And then comes back at noon and knocks on the door one more time and then leaves. If they're too far apart, in the time, let's say I knocked, that's a little bit of excitation. But then when I'm not knocking, it's going to go back to baseline. So every time I'm starting over and over again, also like when you go to work out, right? You work out one day and then you wait 2 weeks. That's not good because then you're starting from scratch every time, right? So it's over time. You have to do it consistently to get a potential, an action potential or an action, right? So those are the types of summation for those graded potentials with their EPSPs and IPSPs. Okay. Another thing with graded potentials, I said, is that they start at the soma, right? So you're going to receive those neurotransmitters at the dendrites, which are the projections of the soma. And in those dendrites, you're going to have receptors. For graded potentials, the channels that you're using in the soma are going to be ligand-gated sodium channels at the soma, right? So you have those ligand-gated sodium channels at the soma, which when they're activated by an excitatory neurotransmitter, they're going to allow sodium in. So I put sodium in, okay? All right, if they're excitatory. So these are my EPSP channels. If they're inhibitory, they don't open sodium channels. They do other things like activate second messengers. But let's say we're going towards an action potential, we're opening those ligand-gated channels on the soma. Once you reach threshold at axon hillock, which would be what? Negative 55, right? Then the action potential is going to occur, and that's going to open voltage-gated sodium channels in the axon, right? They respond to changes in voltage. So once the voltage is high enough, right, it's going to open all the other ones. It's just getting that initial spark. Once that spark is going, everything else is going to go, right? So action potentials, voltage gating sodium channels. Sodium, again, over there, depolarization is going to influx. It's going to go in the cell. So I'll put that in there in a minute. Okay? Once we get to your synapse, I'm going to put synaptic bulb. At the synaptic bulb, right, I have these channels right here, and these are my calcium voltage channels. So when the action potential gets here, the action potential is going to open the calcium voltage channels, and calcium is going to go in to the synaptic bulb. The purpose of calcium going into the synaptic bulb is to help the neurotransmitters that are waiting there to be released into the cleft. So calcium is going to bind to them here and facilitate their exit into your synapse. And let's say this is a muscle. So I have my ACH nicotinic receptor to cause stimulation, right? So calcium helps with that. That's why calcium is so important for contraction of muscle, for neurotransmitters, and for blood clotting. So depolarization is going to be a sodium influx. Right? That's terrible. Let me write that again. Sodium influx at positive 30, sodium voltage channels close. And in repolarization, potassium efflux, right? It's going out, voltage gated potassium channels, right? And that's how the membrane maintains that. Once that potassium's all the way out, right? Those channels, I said they were really slow. They're just really slow to close, so it's going to cause hyperpolarization where we like too much potassium, right? Now we have to bring it back into balance. And to bring it back into balance, we use the sodium, potassium pump. And this, if it's a pump, it's active transport. So don't let anybody tell you that you're not doing anything because you're just studying, right? You're using ATP. You're using brain power. It's spending energy, right? You're spending energy. Okay. So that would be for that stimulation, depolarization, and repolarization. Okay? Any questions on that? Okay, so we use ligand. Now, there are leakage channels, as you saw in your worksheet. There are leakage channels. Leaky, when things are leaking, it's passive. You're not using energy. I don't discuss them too much because it's going to overcomplicate your understanding of this. So I want to make sure you understand this. And then if you want to add leaky channels later, I can explain to you how they work. But generally what happens is your membrane is at -70 because the leaky channels allow potassium to go out of the cell regularly, right? So the leaky channels do allow that. If the leaky channels weren't there, then your cell would be too positive. They would be too close to each other because sodium wouldn't be able to leave. Now, if you take the potassium out of the membrane, for example, out of the cell, and do not replace it with sodium or potassium again, you're going to get a very hyperpolarized cell. So without the potassium, the actual charge would be closer to like -100, 120. So the -70 is because the potassium makes it less negative than it would be otherwise. Okay, so, all right. We talked about resting membrane potentials. The refractory period versus absolute versus relative refractory periods. Okay, so when we're looking at here and in the neuron specifically, when we're looking here, you're going to see that once these action potential, this depolarization has occurred, that point of time underneath this depolarization area, that's going to be an absolute refractory period because the positive 30 is the maximum amount. So at this point, even if I were to put another stimulus, it wouldn't make the reaction any different, okay? So another stimulus is not going to create another action potential. Another action potential is not going to start right here, okay? This period where it's hyperpolarized, In some cases, you can have a relative refractory period where relative refractory period means that if you have a supra threshold stimulus, that can cause another action potential. So super threshold, super strong, right? Sub threshold, below the threshold, that's for graded potentials, local membrane changes, and super threshold stimulus is for when you're in a relative refractory period. If you put another impulse that's strong enough, it will cause a depolarization or another action potential. You see that more in skeletal muscle. So skeletal muscle, once you contract, you can add contraction to it, right? We talked about twitch, trep, and wave summation. So you're able to do that with muscle. Most other things you're really not. Like your heart muscle can't do that, for example. Okay. So what's next? Let's see. I want to go with the flow soon. All right, that's most of chapter 12. Any other questions on chapter 12? No? All right, so next we're going to go into chapter 13, right? And chapter 13 was more of what? The anatomy of the nervous system? Embryological development, right? The anatomy of the nervous system, pretty much where it comes from, etc. Okay? So we're going to be talking about your vesicles, right? Your primary vesicles, secondary vesicles, etc. So when you were an egg and a sperm came together, they formed a zygote. This is an introduction, like a movie introduction, right? Right, somebody needs to put music in the background. So you first were a two-cell being, right? 23 chromosomes for 23 chromosomes. They finished meiosis. When meiosis is finished, they did their first mitosis and became a zygote. From a zygote, you become a group of cells called a blastomere. From there, you become a morula. From there, you become a blastocyst, and from a blastocyst, you become an embryo. Okay? Once you become an embryo, it means you have differentiated cells. So that's when we start our journey. Okay? So in terms of differentiated cells, we're talking about the 3 germ layers. And the 3 germ layers is what you're tested most on in terms of embryology. So that's also known as a trilaminar disc as part of the blastocyst, but we're talking about ectoderm in this chapter, right? Ectoderm, mesoderm, endoderm. Ectoderm. Ectoderm is going to make your nervous system, and it's going to make your integument, right? Which is your skin. So those are the 2 things that the ectoderm starts, right? From that ectoderm, we're going to develop, and we're going to talk about the neural tube, right? You have your neural tube, which will be your spinal cord, neural crest, which is going to be your peripheral nervous system, and then you have your neural plate, which is what's going to become your brain and brain cell. Okay, so from there, we're going to start here with the vesicles. So between 3 and 4 weeks of being an embryo, so of gestation, right? We call that gestation. You're going to have 3 primary vesicles, right? And these vesicles are all going to end in encephalon. What was encephalon? Brain, right? Encephalon means brain. So you're going to start with the prosencephalon. You're going to then have the mesencephalon, and then you're going to have the rhomboencephalon, right? Those are the 3 primary vesicles. You're only those 3 vesicles for a week. Even before your mom knew she was pregnant, that's it. You never find out, okay? So we move on to when you actually find out. So about 5 to 6 weeks, around that time, you're going to divide again into 5 primary vesicles or secondary vesicles, and that's going to be the telencephalon, the diencephalon, the mesencephalon stays mesencephalon, that one doesn't change, and the rhomboencephalon, which becomes the metencephalon, and the myelencephalon. They all have the same last name, they're all related, right? So it's like having children, right? One's tele, one's dia, one's mesa, one's meta, one's myelin, right? Right? Okay, telencephalon. What comes out of the telencephalon? What structure? Telekinesis? Cerebrum, right? Cerebrum. So anything related to the cerebrum, lobes, basal ganglia, ventricles, all that stuff, right? Eye cup too, the eye cup. All right, diencephalon is your anything that ends in thalamus. Anything that ends in thalamus. Hypothalamus, thalamus, epithalamus. Okay? The mesencephalon stays as the midbrain. That does not change. The metencephalon divides into two, and that's going to be your pons and cerebellum. And then your myelencephalon is your medulla oblongata. Okay? So that's where your brain comes from. Primary, secondary vesicles. Okay? All right, so let's talk about each region individually. All right. In terms of the cerebrum is divided into lobes, but it's also divided into the cortex. The cortex is your gray matter. Anything that is not, the cortex, like the inside of the brain, is white matter because the cortex connects to each other. But inside of the cerebrum, you also have the basal ganglia, which are islands of gray matter, right? I said it was a chain of islands. They each have their own name, collectively known as the basal ganglia or basal nuclei, but they each have their own specific names. Kind of like I said Hawaii or the Bahamas, right? I'm going to the Bahamas. Where? Nassau, Freeport, Exuma, right? They all have their own name, okay? They all do different things too. But anyway, we also have lobes, right? We separate it into lobes, and then your basal nuclei or ganglia. Okay? So if we start with the lobes, let's start with the frontal lobe. What are the most important parts of the frontal lobe? Start with the prefrontal cortex, right? That's the one your parents tell you don't have yet. Prefrontal cortex, decision-making, executive function. Executive function. That's one of the last things to form about the age of 25-ish. Some people, it never finishes. But, executive function, right? Decision-making. So 3 decisions you don't make, what were they? Kids, tattoos, and student loans, right? Wait till you're 25. Okay, the next thing that we have in there is the precentral gyrus, right? And a gyrus was the wrinkle, right? The gyri are the wrinkles of the brain. The precentral gyrus did what? Primary motor cortex. Man, let's tie this together with other chapters. Primary motor cortex, your instructions for movement, your conscious instructions for movement. Which tract was in charge of conscious movement? Descending tract that is in charge of conscious movement. No? Not yet, right? Not yet. Okay, that would be your corticospinal tract, right? With your upper motor neuron to your lower motor neuron, right? 2 down, 3 up, right? Descending tract. Primary motor cortex tells me what to do. Remember that these tracts, all these are contralateral. What does that mean? Right? The right side of my brain controls the left side of my body and vice versa. That's what that means. Okay. What another area? You have 1 area, 2 areas. The 3rd area in the frontal lobe is Broca's area, which was speech motor, right? Motor for speech. Important area for the development of the ability to speak. Okay? From there, I'm going to move on. Separated from the frontal lobe by the central sulcus, you're going to have the parietal lobe, okay? And the parietal lobe has something important called the postcentral gyrus. And what does the postcentral gyrus do? Conscious perception, right? Conscious perception, somatosensory cortex. Conscious perception of pressure, pain, temperature, right? Sensations, general senses. Okay, parietal lobe has that. The postcentral gyrus has the somatosensory cortex. So here you're going to receive information. Receives input from general senses, which are the tracts, which are the ascending tracts that we went over. We had the ones that had the first, second, and third order neurons, right? Because it takes 3 neurons to go up and 2 neurons to go down. So these are going to be your dorsal column, right? Dorsal column pathway, also known as the dorsal column medial lemniscus, right? Which was fine touch and proprioception. You're going to have the anterolateral spinothalamic tract, which was pain and crude touch. You're going to have the trigeminothalamic tract, which was sensory for the face. Trigeminal, sensory for the face. Trigeminothalamic tract, sensations from the face. So those are your ascending tracts. And those are ascending tracts. These are conscious, right? Conscious tracts, like you're aware of pain, you're aware of touch, you're aware of someone touching your face. So these ones are going to be all contralateral as well, right? So whatever somebody touches you on the right side, your body interprets it on the left, okay? Unless it's unconscious, in which it would be spinal cerebellar, which is unconscious proprioception, and that is on the same side, which is ipsilateral. Okay? The other lobes, so let's go to the next lobe, temporal lobe. Temporal lobe is going to do a lot of things, actually. Here you have Wernicke's area in the angular gyrus in the back of the temporal lobe between the temporal and the parietal, which is going to be interpretation of speech. So interpretation of speech, in other words, understanding speech. You're going to have the olfactory cortex there. So your smell's going to go there. And you're going to have your auditory cortex there as well, right? Smell goes to the temporal lobe. Auditory cortex goes to the temporal lobe, okay? The olfactory system, you had your olfactory nerve, right, which is cranial nerve 1, going to the bulb, and then going from that bulb to the temporal lobe. And then your auditory cortex, you had your cochlear division of the vestibulocochlear nerve, and that is going to go also into the temporal lobe. So I'm just going to put the origin there. Okay, next, occipital. And occipital, we're just going to generalize it as 1, right, which would be your visual cortex. Visual cortex. The visual cortex is both ipsi and contralateral for vision, and it gets a lot more complicated than that. So just knowing that the visual cortex is the occipital lobe is fine. And you have the limbic lobe. And the limbic lobe is located in the cingulate gyrus, which is right above the corpus callosum, right? And the corpus callosum was the white matter connections between the right hemisphere and the left hemisphere, which are divided by a big crack called the longitudinal fissure, okay? So the limbic lobe cingulate gyrus, this is going to have 2 big structures here, the amygdala and the hippocampus. And if you want to generalize those for now, the amygdala is going to be your emotions to certain things, and the hippocampus is memory, like a lot of the emotional memory. Like we talked about the olfactory nerve, right, going to the thalamus, to the temporal lobe for knowing that you're smelling something, but then it went to the amygdala for emotions about what you smelled, and the hippocampus for memory to see if you remember the smell, and to the reticular formation in the medulla oblongata for visceral reactions to that smell, right? So the autonomic nervous system is going to be hypothalamus, brainstem. That's a lot of your autonomics are autonomic nuclei, like the sensory information from your autonomic nervous system is integrated in the brainstem. Respiration, blood gases, blood pressure, things like that. Okay, so I'm talking mostly conscious things right here. Then you have the basal nuclei, which are a collection of gray matter nuclei in the center of the brain. They surround the thalamus area, like they're on the outside of the brain, 3D, so it's hard to see them. And in that area, you're going to have control of movement. So it's going to help smooth out and control the movements of your body. It's a major inhibitory pathway. So what it does is it mainly prevents you from moving too much. And if it goes out of whack, then you would have tremors and things like that. Okay? So we have those main parts of the brain. The diencephalon. The diencephalon, I said anything that ends in thalamus. So you're having the thalamus itself. The thalamus is your major relay for a lot of pathways in your body. The hypothalamus has nothing to do really with the thalamus. That is going to be homeostasis and the control of the autonomic nervous system. And the epithalamus, the major structure, is going to be the pineal gland. And what hormone do you produce in the pineal gland? Melatonin. Okay? From there, midbrain pons and medulla oblongata are going to make up your brainstem. Like I said, control of your autonomics. The cerebellum is balance and unconscious proprioception. So it keeps track of what your body is doing kind of in the back burner, so you don't have to think about it. So it does that for you as well. I'm not going to go into very many details about those structures. We don't have time to cover everything like that. But in your test, this is majorly what we focus on. And then the major functions of like the cerebellum and things like that. So cerebellum, unconscious proprioception, think spinal cerebellar, think of like unconscious movements, control of unconscious movement and balance, okay? The other part we had balance at was in the vestibule of the auditory system, right? We talked about the vestibule and the semicircular canals, which we'll go over a little bit later. Okay, we did all that. Make sure I don't miss anything here. All right, so let's talk about the structures of the rest of the central nervous system. So I'm going to go ahead and start with your spinal cord, which is what you saw when you walked in, right? You have the spinal cord slice. I don't know why. I like this marker because it's very inky, but it's really hard to erase. So let's start here with that spinal cord cross-section. So what areas of the spinal cord do you need to know? So we had in terms of the major areas of the spinal cord, if you were looking at it in gross anatomy, you have the cervical enlargement, right? Then the lumbar enlargement, the conus medullaris at L1, L2, which is where the spinal cord ends in adults. And then anchoring the spinal cord to the coccyx, you have the filum terminale, right? All the nerves that come after the conus medullaris, we group them together as the cauda equina because it looks like a horse's tail. So that is the gross anatomy of when you look at it, right? When you're looking at it in the models at the lab and things like that. When we're talking about the cross-section of the spinal cord, I'm going to start it over here. I'm going to make it the other way now. So I have my little cross-section here, and it's not going to be perfect, right? I'm not an artist. Okay. So here you have your cross-section. In that cross-section in the back here, you're going to have your dorsal nerve root ganglion. In the front, you have your ventral root. They come together, and they form a little area here, which is your spinal nerve. Your spinal nerve does separate again. They'll separate again here. Okay, that look right? All right, so here we have the area, right? Dorsal horn, lateral horn, imagine it's there, and then ventral horn, right? The dorsal, anything dorsal is going to be sensory. So sensation comes in through the dorsal root, dorsal nerve root ganglion, and dorsal horn. That's sensory information. Motor information, if it's autonomic or if it's somatic, skeletal muscle, it doesn't matter. It's going to go from the lateral or ventral horn, depending, right? Lateral horn is autonomic. Ventral horn is somatic nervous system, and it's going to go out the ventral root, right? So you have your root, ventral root, dorsal nerve root ganglion, DNRG, okay? Once they go out far enough, there's going to be an opening right here. That is your intervertebral foramen, right? At that point, they go together, and now I have sensory and motor fibers at the same time. So that would be called your spinal nerve, right? Because spinal nerves are mixed nerves, meaning they have sensory and motor, okay? From there, it does a couple other divisions. Here you have your dorsal rami or ramus, your anterior rami, and here you have your white and gray rami communicantes, because they communicate, right? All right, out here you see you have another little ball. That is your paravertebral ganglion. And you know that if it's a paravertebral ganglion, you're assuming this is autonomic nervous system, okay? The anterior and dorsal rami are part of the somatic nervous system, which is going to be skeletal motor and skeletal sensory. Okay, so those are that cross section of the spinal cord. These white areas are the columns, and in those columns, these white matter areas, is where we have your tracts, right? So that's where you have your tracts. So we talk posterior column tract, or dorsal column, right? We talked about fine touch and proprioception. Anterolateral, it's going to be an anterolateral tract, which is spinal thalamic. So it's telling you where does it go up. So if sensory information comes in through the back and it's proprioception, it's going to go through that posterior column up to the brain, right? For example. So the columns are the elevators for the sensory or motor information. Okay. From there, I already told you the big areas of that spinal cord. Let's talk about the brain again. I kind of miss a couple things here. So that's anatomy. Anatomy, just bare anatomy. Let's talk about the other parts of the brain that I missed. So going back to the brain. The brain has cerebrospinal fluid. So we have to produce it somewhere. So in terms of the cerebrospinal fluid, or your CSF, you have your ventricles. How many ventricles do we have? 4, right? You have 2 lateral ventricles. The lateral ventricles are separated by a thin membrane called the septum pellucidum, just in case you were curious about that. Okay, so you have 2 lateral ventricles. They are connected to the third ventricle by the interventricular foramen, also known as the foramen of Monroe. Okay, that third ventricle is going to be connected via the cerebral aqueduct to the 4th ventricle, and the 4th ventricle is located between the pons and the cerebellum. Okay, the 3rd ventricle overlaps the area of the thalamus, so you will see that it's kind of medial, right? Because it's in the center, and then the thalamus is a little bit on the side, and then the basal ganglia is on the side of that. And the lateral ventricles are under the corpus callosum. There will be a cavity there. The inside of the lateral ventricles, you will notice sometimes that there's like a grainy tissue in there, and that's called the choroid plexus, and that's where you have a concentration of ependymal cells. So I actually do have human brains that have that, and I can show them to you in the lab if you want to. I found them last week. I was digging in there and found the choroid plexus. All right. CSF. Now, CSF does get reabsorbed into the body. It gets reabsorbed as part of your venous blood, right? Deoxygenated blood. The brain itself, it does have veins, but major venous drainage is going to happen via the sinuses. So to look at the sinuses, first we have to look at the meninges. The meninges are the connective tissue coverings of the brain. And first one you have is your dura mater, which means hard mother, right? Hard mother. So that is the outermost covering. And in that dura mater, you're going to have embedded your sinuses, your cerebral sinuses, right? Which are going to collect venous blood. And CSF from the arachnoid granulations. Okay? Collect venous blood from the arachnoid granulations. So that's dura mater. Underneath the dura mater, you have the subdural space, and underneath that, you have the arachnoid. Arachnoid because it's spider web-like, right? Arachnoid matter. And the most important aspect of this is where you're going to have your subarachnoid space. And the subarachnoid space is where you hold a lot of that CSF flow. So I said the CSF goes from lateral ventricles, third ventricle, cerebral aqueduct, fourth ventricle. From there, it goes through the central canal. The central canal was that little hole here. Central canal in the spinal cord. And it goes there, and then it comes around, and it goes through the subarachnoid space all the way up, and then it gets reabsorbed into the sinuses via the arachnoid granulations, okay? Then the smallest one that you have in terms of meninges is the pia mater, which means small mother, and that one just anchors vasculature to brain tissue. I say brain, but I mean like central nervous system tissue. Your central nervous system has a lot of blood vessels. They need to be anchored even in the spinal cord. You have like an anterior spinal artery, a posterior spinal artery. So it's very highly vascularized. We don't show it that much because it's just a lot, like the more we dig, the more we find kind of thing. Okay, so I'm just showing you the main parts. Okay, so now we know we have the CSF. The CSF goes around, right, subarachnoid space, gets reabsorbed into the venous blood. That venous blood is going to empty into a vein called the internal jugular vein. The internal jugular vein runs alongside of the internal carotid artery. Most of your veins have the same name, like renal artery, renal vein, but in this case, they just decided to do that. Okay, now vasculature. Vasculature of the brain. Vasculature of the brain. We're talking about the circle of Willis. Right? The circle of Willis. There's two main arteries that feed the brain. The one at the bottom here, coming together. Oh, I'm going to have to erase that. That was too big. Right? You have your vertebral arteries. Right? Two vertebral arteries. One from the right subclavian, one from the left subclavian. They're going to go in here through the foramen magnum. And what bone was the foramen magnum? Occipital. Occipital bone, right? And they're going to come together and form this thick artery that is known as the basilar artery. And you're going to see it right on top of the pons, of medulla and pons. So I'm going to put your brain stem. Okay? Let me erase this real quick so I can keep going. All right. Now here, coming through the carotid canal, I have my internal carotid. My internal carotid has 3 branches. It's going to branch down here. It's going to branch down here, right? It's going to branch out here. And it's going to branch towards the front like this. Right? And it has a little connection here. Okay? So this is your internal carotid. It's been cut. That's why it's a circle. It's like cut it. Otherwise, it would be hanging this way, right? So here you have this one, which is the posterior communicating artery. Communicating artery. This one right here goes to your parietal lobe. That's the MCA or middle cerebral. That's not part of the circle, but it's there. And then you have these two going out the front, which are the anterior cerebral arteries. And then this bridge that connects the right side to the left side, and that is the anterior communicating. And that's your circle of Willis. That is the main vascularization of the brain. You'll notice that I only put the main arteries there. When you look at the circle of Willis in real life, you have arteries that come off here, off the basilar. Those are pontine arteries that feed the pons. You have two arteries that come up here, the posterior superior cerebellar and the posterior cerebral artery. Then you have your posterior communicatings, etc., etc. So there's more arteries that come from there, but those are the main ones for the circle of Willis. All right, I covered all of that. Let's move on to the little bit of that peripheral nervous system. Okay? So, in the peripheral nervous system, we're talking about, remember, I'm just going to summarize this one more time. What is a collection of cell bodies in the peripheral nervous system called? Collection of somas. Cell bodies in the central nervous system, nuclei. Axons are tracts, right? So in the peripheral nervous system, the bodies are ganglia, and the axons are nerves, right? Remember that nerves are made up of many neurons. Neurons are the individual cells, so they are arranged into fascicles. Because they're arranged into fascicles, we can have cables coming and going inside of the same nerve. The entire nerve is covered by epineuron. The fascicles are covered by perineuron, and the individual neuron is endoneuron, just like the muscles, right? Kind of like the same thing. All right. From the peripheral nervous system, we're looking at anything that exits after that intervertebral foramen. So in the peripheral nervous system, we have two main kinds of nerves. You have spinal nerves, and cranial nerves, right? Cranial nerves. Your spinal nerves are going to be mixed nerves, meaning they have sensory and motor, and they are mainly formed from anterior rami, right? Anterior rami. When those anterior rami come together, we make our plexuses, or plexi, I don't know, plexus. All right, remember from the cervical enlargement, we have the cervical plexus, and we have the brachial plexus. The cervical plexus is more of that sensory and motor information for the muscles of the anterior neck and muscles in suprahyoid, infrahyoid, scalenes, right? Things like that. Cricothyroid, thyroid. Okay. But it also has one more nerve, right? So it goes cervical plexus from C1 to C5, right? And the main nerve that I taught you from here was from C3 to C5 roots. And what was it called? No? Cervical plexus. What was the main nerve off the cervical plexus? Phrenic diaphragm, right? Phrenic nerve. Voluntary control of that diaphragm, your main breathing muscle. Because paralyzed during polio, that's how kids ended up in those iron lungs. Okay? Because the poliovirus attacks your neurons. All right, upper limb, brachial plexus, C5 to T1. Remember that in terms of nerves or nerve roots, we have 8 cervicals. Cervical is 1 through 8, right? So we have a C8 nerve. We don't have a C8 bone, but we do have a C8 nerve. So upper limb here, we're talking about those main cords at the end of them, because they mix together from roots, trunks, divisions, then cords, right? So in the cords, you're going to have the musculocutaneous nerve. Musculocutaneous nerve is going to be mainly C5, 6. You don't have to know those details, right? But it innervates everything that has to do with the biceps. Biceps brachii, brachialis, brachioradialis, coracobrachialis, etc. All right. From there, in the lateral forearm, you're going to have the median nerve. And the median nerve is it comes from all the levels. It's a union of all the roots. But we generalize it as C7. And it's going to be lateral flexors. Lateral flexors, right? If there's paralysis of the median nerve, I just say if there's compression, it's carpal tunnel. But if there's paralysis, it's a condition called ape hand, like this. Okay, so if you see ape hand, it's median nerve. Okay, on the medial side, you have ulnar nerve. And ulnar nerve, those medial flexors, and it does a little bit of sensory in the back of the palm too, like back and front. But I'll just leave it as medial flexors. And it's roots from C8 to T1. So that's the bottom there. All right, from the posterior cord, you're going to have the radial nerve. The radial nerve is going to do the extensors of the forearm. And then you have the axillary, which is C7 again. And that one, this is C6. And axillary nerve is going to do deltoid and triceps. Okay? Then the thoracics, right? The thoracics don't form a plexus. They just do intercostal nerves, because most of the nerves from that thoracic spine are going to become part of the sympathetic ganglia. But the anterior rami of the thoracic spine just does the intercostal nerve, so we don't have a thoracic plexus. Okay? This is just the upper limb part, but in the trunks and stuff of that brachial plexus, you have the nerves that go to your pecs and like the muscles in that area. And from the lumbar plexus, you have the ones that innervate your abdominal muscles. We just don't go into those details necessarily right now. Okay? Lumbar plexus. Lumbar plexus. That is L1 to L5. Some books will argue that is up to L4, but I'm just going to say L5. Okay? From there, we're going to get 2 main nerves. Femoral nerve. And femoral goes to your quads. And then we're going to have obturator. And obturator goes to your adductors. Okay? And then lastly, sacral plexus. Sacral plexus includes a little bit of that lumbar trunk, L4, 5 to S4, depending on which book you read. And the main nerve out of that one is your sciatic. You also have pudendal, but sciatic, and the sciatic is made up of 2 nerves, the common peroneal or fibular, common peroneal fibular nerve, and the tibial nerve. Huh? It's going to innervate hamstrings and the entirety of your lower leg, back and front. That is the plexus. Okay? Then you have your cranial nerves. We went over the cranial nerves, I think, a lot. So if you guys want to go over that, I'll do it later. If anybody has questions on the cranial nerves. I am going to move on to the pathways. Okay? So in terms of the pathways, we had 2 chapters on that. Chapter 14. Chapter 14 was somatic nervous system sensory. So ascending pathways, which I went over, first, second, and third order neurons, right? 3 neurons to go up. Anything sensory takes at least 3 neurons to go up. So if we're talking somatic sensations, we're talking about mainly general senses. We did talk of special senses when we talked about the cranial nerves, but general senses, which are gonna be either encapsulated, right? Sensory receptors like Pacinian, Merkel, Ruffini, or Meissner, or it's going to be free nerve endings. Free nerve endings are mainly pain, nociceptors, and thermoreceptors, okay? But your pressure, vibration, all of those are generally encapsulated. If they have a name, they're encapsulated. If they have free nerve endings, we just call them free nerve endings, okay? In terms of those pathways, we were talking about 2 ascending, right? Ascending pathways. And we were talking about dorsal column, right? Dorsal column and anterolateral or spinal thalamic. Spinal thalamic. Okay? The dorsal column is going to be fine touch and proprioception. All right, proprio here. And spinal thalamic, anterior, is going to be crude touch, mainly, and lateral is going to be pain and temperature, right? Because if you remember, if the temperature is too cold or too hot, instead of the thermoreceptors reacting, you're going to get pain receptors instead. Too cold, pain. Too hot, pain, right? Pain is a slow adapting receptor so that you don't hurt yourself. The dorsal column receptors are more, especially these fine touch ones, like putting on your clothes. Those are fast adapting because you don't need to know that you're wearing clothes all the time. I mean, you do, subconsciously. But you don't need to be consciously aware that you're wearing clothes, I guess. I don't know. And then proprioception are slow adapting because if you're in a situation where your body is at an angle that is dangerous, your body shouldn't be getting used to it any quickly. The dorsal column and the anterolateral system both start with a peripheral nerve. And when I'm talking about peripheral nerves, I'm talking about just one of those nerves, the ones that we can name really well, right? So from peripheral nerve, it's going to go through the dorsal root, right? From that dorsal root, it's going to go into the dorsal horn. And from the dorsal horn, it's going to go to the dorsal column. From the dorsal column, it's going to take the elevator to the medulla. At the medulla, it's going to cross over to the opposite side of the thalamus. And from the thalamus, it's going to go to the somatosensory cortex in the parietal lobe, right? In terms of the neurons, your first neuron, your peripheral nerve, is going to go from here to the medulla. At the medulla, it's going to meet the other nerve, which is the one that's going to cross over to the thalamus, and then from the thalamus to the somatosensory cortex, first, second, third order neuron, okay? The spinal thalamic tract, I'm going to put here, peripheral nervous system, right? Dorsal, everything, dorsal, remember the dorsal horn. But instead of going from the dorsal horn to the dorsal column, instead it's going to go to the anterior or the lateral column, depending on what it is. And from there, no, my bad, dorsal horn. At the dorsal horn, it's going to cross over. Decusates to opposite antero, I'm going to just put this together, anterolateral columns. From there, it's going to go to the thalamus, and from the thalamus to the opposite side, somatosensory cortex. So they end up in the same place, but this one crosses over immediately at the spinal cord, and the other one crosses over at the medulla oblongata. Those are the main 2 pathways of the general senses of your body. Spinal cerebellar is unconscious, but it's on the same side. And trigeminal thalamic does both sensory and pain from your face, but it crosses over immediately at the pons and the medulla to thalamus to the opposite side. Okay? Those are the main ones for those general senses. Everything else is motor. Okay? If you guys need to go, you can. I know it's 12:15. I just want to go ahead and finish as much as I can here. All right. So I'm going to go down the motor pathways. Okay? So this all general senses, this is SNS, right? Somatic nervous system. If we're going to go to descending pathways, I'm going to start real quick with the descending pathway for your somatic nervous system. And this is only going to take me like maybe 10 minutes. Somatic motor. Somatic motor means skeletal muscle. Okay? So going 3 up, 2 down, right? 2 neurons to go down. So you're going to have, in this case, you're going to have an upper motor neuron, and that's going to go to a lower motor neuron. This pathway crosses over at the medullary pyramids, right, which were the columns of the medulla oblongata. So it is contralateral. The right side of your brain controls the left side of your body. So these upper motor neuron is a central nervous system neuron. Right, so this would be a tract that is going to connect at the spinal cord with a lower motor neuron, which would be your peripheral nerve that we know and name, right? Sciatic, axillary, etc. Okay? What is the name of this pathway? Where is it going from? Where's your primary motor cortex? Yeah, so it goes from the primary motor cortex in the frontal lobe to the spinal cord. So it's going to be cortico-spinal tract. Cortex to spine, cortico-spinal tract. So this is your cortico-spinal tract. You have one long-ass neuron that goes like this. It crosses over, it goes all the way down, and then, right? So it's one long neuron. That is the main of that. You also have the cortico-bulbar tract, but that is head and neck. Head and neck movement, right? Head and neck motor. Okay? But the rest of your body is cortical-spinal. There's an anterior and a lateral. I'm teaching you the building blocks of these pathways, not the entire pathway. Okay? So when I show this video to my friends, when they see it, they're going to be like, that is not entirely correct. It is correct enough, okay? Correct enough. All right, so let's talk about autonomic, and we're done. Okay? ANS. The autonomic nervous system. Right? Autonomic nervous system is controlled by hypothalamus. Hypothalamus is homeostasis. It does a good job because it controls glands, smooth muscle, and cardiac muscle. Okay? Anything involuntary, right? In the autonomic nervous system, as you have over there, the neurons of the autonomic nervous system come out of the lateral horn and it go to the ventral root. They go out the white ramus and they go to a ganglion. If you must know the pathway of that. Okay? The autonomic nervous system has 2 parts, the sympathetic and the parasympathetic. The sympathetic is fight or flight, and your parasympathetic is rest and digest, right? The sympathetic comes out, the nerves come out of the thoracolumbar spine, thoracolumbar, fight or flight, parasympathetic, craniosacral. That is the origin of those nerves and those areas. Okay? The sympathetic nervous system is going to have a short preganglionic neuron and a long postganglionic neuron most of the time. And it's going to end up, right, at your effector. I don't know if you guys can see that back there, but I'll put it here. I won't erase it. At your effector, or it can have a preganglionic neuron going to everybody's favorite gland. You'll notice that I said about everything, right? Medulla, has a medulla, which is the adrenal gland, right? Adrenal gland, adrenaline, right? Think about it like that for now. The parasympathetic nervous system is going to have a long preganglionic neuron and a short postganglionic neuron ending in your effector, whatever that is, right? That preganglionic neuron on both sides, pre, is going to be lightly myelinated. Lightly, right? Sprinkled like sugar. B fiber, right? B fiber. Those are B fibers, right? B fiber, B fiber, B fiber. They're all B fibers, which means they're lightly myelinated, as opposed to these ones. These are all A fibers. They're all A grade fibers, the best, the fastest, the fattest. Okay. In this case, if you're big and fat, you're fatter, you're fast in somatic nervous system. In the autonomic nervous system, the B fibers are a lot slower, but they're still doing saltatory conduction, because if they're myelinated, you're jumping from notoramvir to notoramvir. Remember that? Okay. The postganglionic neurons, post, are going to be C fibers. C fibers, where the unmyelinated fibers, right? So they're slower, and C is going to have continuous conduction. So they're a lot slower. Okay? Now, they're called pre- and postganglionic because they're going to synapse at a ganglion. In the case of the sympathetic nervous system, they're going to synapse at a ganglion that we called sympathetic chain ganglia, aka sympathetic trunk, aka paravertebral ganglion, because they're on either side of the spinal cord, and they're found here, right outside of the spinal cord. So these, when I put paravertebral ganglia, they're right outside of the spinal cord, and that's usually where they synapse, except for three other ones, right? The three ones that go to the enteric nervous system, which are going to be the celiac ganglion, the superior mesenteric, and the inferior mesenteric ganglions. Those are going to be prevertebral ganglions because they're not next to the spinal cord, they're in front. Okay? In terms of the parasympathetic, we have terminal ganglia because they are close to the target organ. The parasympathetic nervous system, the cranial part is going to be cranial nerves 3, 7, 9, and 10, and sacral segments S2 to S4. The thoracolumbar spine is going to be T1 to L2 because the spinal cord ends at L2. Okay? So, in terms of neurotransmitters, the first neurotransmitter here to come out, it's going to be your acetylcholine, right? Acetylcholine, right? Acetylcholine, and acetylcholine into the muscle, right? All of these red acetylcholines are going to be nicotinic. You know, you got the drift, right? And for nicotinic. Okay. Nicotinic receptors. Remember those? Nicotine makes you alert, right? Nicotinic receptors. It causes stimulation. The nicotinic receptors are ligand gated channels. That's why they're excitatory. Okay, that's the difference. Now, in the sympathetic nervous system, the other neurotransmitter that we're going to secrete at the end of it all is going to be norepinephrine and epinephrine into adrenergic receptors. In this case, the acetylcholine is going to stimulate the medulla of the adrenal gland to secrete norepinephrine and epinephrine into the bloodstream so it can act as a hormone and stay in the system a little longer. In the case of the parasympathetic nervous system, we're going to secrete acetylcholine again. But in this case, this acetylcholine is going to go into muscarinic receptors, which are the resting and digesting ones. So in this one case, acetylcholine is inhibitory because it's who receives the gift, not what the gift is. In terms of those receptors, right? Those receptors, we can go ahead and generalize them, place them into categories. You're going to have the ones for acetylcholine, which are called cholinergic, and that is going to be nicotinic, which is ligand gated, which is going to be excitatory, or you're going to have muscarinic, which is going to be a GPCR, G protein coupled receptor, which is going to be inhibitory. In case of the noradrenaline and adrenaline, you have the adrenergic receptors, right? And those are going to be alpha or beta, alpha 1, alpha 2, or beta 1, beta 2, and beta 3, right? So those are going to be your main receptors. Alpha 1 is going to be more of vasoconstriction, if I'm not mistaken. I'm more familiar with the betas. Increased heart rate, increased bronchodilation, right? Think of albuterol. And B3 lipolysis, which is going to be breaking down fat for energy. Those are all norepinephrine receptors, and they're always excitatory in some way, shape, or form. They're always sympathetic. Okay? Yeah. Any questions? No? Okay. So that's all I got for you guys today right now. If you have any other questions, please reach out to me. I'm going to do another review tomorrow, so I'll review it one more time tomorrow with one more class that I have, and that will be that for that. I did record it. The recording doesn't follow me really well. It did better than it usually does, but it's not great. But at least you have the sound if you want it. So I'll go ahead and upload that as soon as I have the file.